Hello and good afternoon, San Diego time. I'm very happy to be here and I'm thankful to the conference organizer that have invited me. I like this conference very much and I'm happy to be presenting here today. So let me just share the screen and I will start. I want to tell you today about special samples for obtaining non-invasively the inner colonic bacterial communities of the human gut. I also want to talk to you about how unique and profoundly different they are from stool samples. But before we dive into the clinical trials and results, I want to introduce you to the Hygieacare system for colon irrigation. This is an FDA-cleared Class 2 medical device, prescription only, where the indications for use is when medically indicated, such as before radiological or endoscopic examination. The Hygiprep is a simpler and easier bowel prep for colonoscopy. It actually replaces the need for an oral purgative. So far, we've had over 14,000 procedures. We have done over 14,000 procedures, 97% clinical adequacy. The procedure has been proven to be safe, fast, easy, and convenient. We have an excellent safety profile and an excellent patient satisfaction. The Hygiprep is agnostic to poor predictors for colonoscopy bowel preparation, including diseases like IBD, IBS, diabetes, and chronic constipation. It is also proven to be suitable for physically impaired patients that can enjoy this prep. And with working together with the gastroenterologist and upon signing consent, we have access to patients' medical records that reflect the prevalence of all disease in the colorectal cancer screening population. Our headquarters are in Texas, in Dallas, Texas. We have centers in Austin, Texas, Cincinnati, Ohio, Jackson, Mississippi, Norfolk, Virginia, and Rockville, Maryland. We also have a US rollout process with a significant pipeline nationwide. Here is word cloud analysis of patient's response, open text response for oral prep and Hygiprep. This was done in 2 of our centers, and as you can see, the difference is dramatic. Over 1,000 patients that have had an oral prep didn't really like it. The size of the word is correlated to the word abundance in the open text answers. And on the right-hand side, you can see the response of patient to the Hygiprep. So very, very dramatic difference. Patients like the Hygiprep very much. And it is for many of them the preferred bowel prep for colonoscopy. For the gastroenterologists in the crowd, you can see some images of colon after the Hygiprep. It was given to us by Dr. Stessen from Austin Gastroenterology. You can see that the colon is clean and ready for scoping. And now I would like to switch gears to the Hygisample, a unique non-invasive method to obtain inner colonic microbiome samples. We're all here because we understand the potential of the microbiome. We know that the majority of known disease can be associated and or reflected in the composition and function of the gut microbiome. And we know, and I believe that most of us believe, that when modifying the gut microbiome and really understanding what is the functional dysbiosis of different disease from neurologic to oncologic, we will be able to significantly improve and completely change the way we look at medicine, personalized medicine, drug design, biomarker, so on and so forth. We at Hygieacare believe that our access to screening population will allow for collection of colon effluent for all disease indications and really have a pivotal role in moving this paradigm shift forward. Another aspect of the Hygie samples collected during the Hygiprep is the fact that they're biogeographically relevant. In the past 15 years, there have been a huge body of literature showing how many biogeographical environment we can find in the colon, right? We have the rectum and the left colon and the transverse and the right is connected to the small intestine. And most of what we find in literature is being done in stool samples, which are a proxy, but the question is how far the proxy is from what there is really in the colon. The other body of literature that we find on microbiome, gut microbiome data is from biopsy samples from gut that was prepped with an oral prep that also has issues because it actually cleans the gut mucosa and it modifies the microbiome. So the Hygie sample is a patented method for microbiome sampling and analysis. And we believe that it provides a non-invasive method for getting a biogeographically relevant microbiome profiling without the effect of the oral purgative. And our ability to collect large number of samples from screening and targeted population can really be pivotal for the field. Also, the fact that samples are being sampled in our centers by trained personnel in a controlled environment. So we have nurses and techs that are trained using standard operating procedure in a very controlled environment. So no longer a person that is sampling their stool at home in a toilet that I don't know how it's cleaned. I do not have SOPs to cleaning in my house. I assume most people do not have it in their own house. Samples can be sent from the different centers to one central lab for standardized analysis. And also, another advantage of the Hygie sample that we have not talked about yet is sampling the missing samples. So what are the missing samples? Missing samples are samples that by accounting for people sampling their stool at home, usually home, we will miss. This could be because of the gross factor, that people are just reluctant to sampling their stool at home. It could also be because several targeted populations have anxiety that is related to bowel function. It could be IBD, IBS patient, constipation patient. Those kind of patients very often has issues, very understandable. So by coming to Hygieacare, that as we said, the Hygie prep is agnostic to those poor indications. We can actually release those patients for the need to sample their stool. And just by signing consent, we can collect their colon effluent. So we have done a study, clinical study. The PI was Dr. David Johnson from Norfolk, Virginia, that as always has been instrumental for us. So the goals of the study were to identify genomic related differences between Hygie sample and stool samples, and to elucidate the correlation between the phylogeny and the sampling time duration of the Hygie prep. We have given our patients the standard DNA Genotek OmniGut tube to collect at home. And then we collected their samples during the Hygie prep. We started with the first. We analyzed the first, middle, and last samples. So we had up to 19 healthy screening population patients. And when I'm saying healthy, they're healthy. No prevalent disease, no underlying disease, healthy. So for each patient, we had 4 samples, 1 stool sample, and 3 Hygie samples. We have looked at different kind of analysis to really verify across the board that we get what we think we get. So we have used microbiome insight sequencing for 16SV for amplicon sequencing to sequence the samples from the first 11 patients. And we used amplicon sequencing variant ASV analysis for that. We also did GIP sequencing 14 million short reads using Sigmatics, Sigmatic in California, that did the metagenomic sequencing for us. For this, we done taxonomic analysis. We also used this data to do BGCs analysis, biosynthetic gene clusters that was done by our collaborators, Vestbiome. This was done on 19 patients, all the metagenomics work. So I want to start with looking at some results. I'll go back to the experimental design for a sec. Excuse me, please. We will start with the home collected stool sample here. So people sampled using the standardized kit with stabilization buffer at home. Then samples were probably collected from the rectum and, maybe here. Then they come and did the hygiene prep. During the hygiene prep, we collected the first, second, and third. You can see the color gradient, it's very significant. For the first two patients, we collected 2 samples here, then we just started collecting 1 per each. Importantly, the last sample that is a proxy, so this is a proxy of the left colon, transverse colon, and right colon. This is where we're actually connecting to the small intestine. And the last sample was always the whitish mucosa sample that is very, very similar to identical to the effluent of the small intestine. After that, only water came out, and it was pretty clear that this is a sample from the right colon. So looking at the alpha diversity by Shannon index, you can see here that the diversity goes down when you go from stool sample to hygiene sample. This is the combined hygiene sample, 1, 2, and 3 together, and this is 1, 2, and 3. Look at this color. It's going to be the same color trend for the rest of the presentation. So you can see here that the diversity goes, but the standard deviation, the diversity goes down on average, but the standard deviation goes up. And we can see that, we can see more about that shortly. Looking at the beta diversity with PICO analysis using weighted and unweighted unifrac, again, 11 patients. You can see on the left-hand side stool versus hygiene sample combined, and on the right-hand side stool versus the different hygiene sample. You can see that when you look at stool and hygiene sample, some phylogenetic similarities from the phylogenetic distance on the multivariate space, and some are more distant from the stool sample. Altogether, stool sample and hygiene sample, but actually basically most of hygiene sample explain about 50% of the phylogenetic diversity of the system when looking at the weighted unifrac. The same trend is also shown in the unweighted unifrac. When you look at the different samples, you can see that there is a shift. When you go towards the left, the right colon, and maybe even the transverse, there is a very big phylogenetic shift based on the beta diversity. We can also see it here. So what we did here, we took the 1,099 species, bacterial species that we found in our ASV analysis, and we did a Venn diagram just by the presence or absence of a specific bacteria. So we did a Venn diagram to look at the unique species found in home collected stool sample and the unique species found in hygiene sample. This is a unified group of species out of the 1,900 that were found in both sample types. This accounts for about half of the species in the system, which is good. Out of the 176 that were found only in stool samples, 77% were Firmicutes, and most of them were Clostridia. As we go more towards the hygiene sample, we can see that the species found in hygiene sample, 31% of all the species found were found only in hygiene sample. But when we look at the phylogenetic trends, we can see that the Firmicutes here are only 58%, where the proteobacteria really are up here. Proteobacteria, many of them are pathogens. The bacteroidetes are about the same amount-ish, but importantly, 15% of the species found here are from unique phyla that are very often not found in those in stool sample. And this, we're going to talk about that again later. Here I show a Venn diagram that shows stool sample, hygiene sample 1, hygiene sample 2, and hygiene sample 3. These are the same 579 and 176. Importantly, though, 2 things. One is that there are some unique bacteria that are found in hygiene sample for all colon segments. which is really important. There is some core bacteria that are found only in Hygie sample and not in stool sample. However, there is also a contribution of each and every of the colon segment for the Hygie sample. And I can tell you that I've looked individually at all the patients, all the 11 patients, and this trend occurs in all of them. Each and every one of the colon segment has a different contribution of bacteria. Now I want to go to the metagenomic analysis, and I want to say that this was done in a collaboration with 8th Microbiome, a wonderful group of people that are working to do deep sequencing, deep genomic sequencing and synthetic biology for discovery of biomarkers and therapeutics. And they work to uncover the relationship between microbial genes and metabolites with phenotypic outcomes. They actually look at gut microbiome to find new biomarkers and therapeutics. They build the VBX1 engine that basically looks at network of microbial genetic blueprints to find gene clusters. And by looking at those gene clusters, they are taxonomic agnostic and are looking at the underlying biochemistry of specific indications. So while they do have the VBX1 engine, they were kind enough to help with taxonomic analysis. So again, we look at alpha diversity using the Shannon index, and we see the same exact trend where the basically the average goes down, the diversity goes down, and it is statistically different. But when you look at the multivariate space with beta diversity, you're using the Bray-Curtis analysis here. Again, we can see that almost 60% of the system diversity is explained by the Hygie sample, and how stool sample clusters so very differently than the Hygie sample. We can also see the gradient where the stool sample is here, the left colon samples are here, and the transverse and right colon samples really go to the other side of the graph. Here is the differential abundance volcano plot of the differential abundance of different Hygie sample over stool sample. So again, going up the column from Hygie sample 1 over stool, Hygie sample 2 over stool, and Hygie sample 3 over stool, we can see that we have samples that are depleted in this comparison, but we have many enriched species as we go farther up the column and compare it to stool sample. Another way to look at that, here you can see this is the Hygie sample 1 to stool, 2 to stool, and 3 to stool. Here is the 0, here is the 0, and here is the 0. And when I do a graphic representation of the Hygie sample 1 over stool, 2 over stool, and 3 over stool with some kind of size relevant Venn diagram, you can see that as we go further from the sigmoid, from the rectum, we can actually get a bigger differential abundance of gene. Again, importantly, there are 15 bacteria. There are 50 bacteria that are differentially expressed between Hygie sample and stool, but they are shared between all the 3 comparisons. So there is some core of bacteria that you can find only in Hygie sample or Hygie sample, but not in stool sample. Then to the BGC analysis. So when we look at the BGC analysis, we can find that as it seems for BGCs, the unified group here is much smaller than with the 16S Venn diagram. However, the BGCs that are found only in Hygie sample are much more pronounced when we look at BGCs versus 16S genes. And I believe that the reason for that is that the phylogenetic diversity of the Hygie sample is much bigger than of the stool sample. We have the biochemical potential is much bigger because we have representation from different phyla. And again, you can see that all the different segments of the colon have a unique contribution to those BGCs found in the different segments. Lastly, this is something that I'm presenting here. This is something we started toying with. We again put the samples on the analysis, the metagenomic analysis, based on taxonomy, same taxonomy I showed earlier here in the multivariable space. And you can see here again how stool samples are really far apart from those samples and how the Hygiesample 1 kind of goes in between. We looked at the phylogenetic footprint, if you will, of carcinoma samples taken from biopsy samples of the left colon. This was taken from a database. These are not our samples. But when we look at that and put it on the multivariable space, we can definitely see here that those samples are more aligned with the Hygiesample rather than the stool sample. And this is something we're still investigating, but I think it really raises the question of whether we should look at stool sample as baseline for clinical indications of microbiome. The last result that I'm going to show you here is that when looking at the 15 most differentially expressed bacteria that we found in the metagenomic sequencing, we have found that 8 were related to biofilm and mucosa. So we have found Firmicutes and Protobacteria, we have found Mycoplasma, we have found here one species of Stachybotrys, another one species, 3 species of Beta Protobacteria, and again, as I said, Mycoplasma. I find it very exciting because I think that this is a very good indication that our samples are enriched in mucosa-related samples that are often absent from stool sample. And with that, I would like to go to the study conclusion. So microbial community structure of Hygiesample is significantly different than of stool sample. I think I've said that. Approximately a third of all bacteria species found were detected only in Hygiesample. 69% of all BGCs found were detected only in Hygiesample, which actually infers a very large biochemical potential of those samples. Each Hygiesample 1 to 3 had a unique species and BGC's contribution. There is an increase in metabolic richness when progressing towards the small intestine. And however, there is a core of differentially expressed bacterial genes shared between all Hygiesamples. The phylogenetic progression and diversity suggest a spectra of individual microbiome, possibly leading to personal medicine, and taxa enriched in Hygiesample overlap with taxa known to reside in biofilm and mucosa layer. Therefore, Hygiesample protected by US patent are 1, the only non-invasive way to obtain intercolonic microbiome, 2, key for defining gut dysbiosis, advanced microbiome-based diagnosis, and AI-based development of biomarkers and therapeutic. And with that, I would like to thank my collaborators. I would like to thank Dr. Dave Johnson, the PI of those studies, and a huge help always from the Eastern Virginia School of Medicine. I would like to thank my colleague, Donato Giovanelli, the PI of the Giovanelli Lab in the University of Naples, for the ASV analysis that he helped us do on the sample sequence by Microbiome Insight. I would like to thank Sigmatic for their great effort in doing our metagenome analysis, and to Vestbiome for all their collaborative work. And with that, I'll be happy to take any question you have. Thank you.